PhD student. I'll start over again. So today I'm going to talk to you a little bit about some research that I've been sort of ongoing since I was a PhD student and continues to this day. The title of my talk is Bacterial Growth Efficiency and the Role of Metabolic Tradeoffs. And below you can find a bunch of contact information for me. Put this aside. All right, so the central theme in my lab is really to understand how factors such as environmental gradients alter the composition and physiology of microbial communities in ways that change their function and their ecosystem processes. So I really try to think about this as sort of this integrated framework that encompasses all the various projects that happen in my lab. The goal really is to link the taxonomic, phylogenetic, and functional diversity of communities to their ecosystem processes. I really take a sort of global change and large ecosystem view of my research, and I center all of my questions along this sort of what are the implications at the ecosystem level. But as you may know, most of my research focuses at the small organism level. So thinking about the role of individual cells and taxonomic groups of bacteria and the role they play in that ecosystem. So to do this, I often ask questions like, how does the physiology of individual bacteria or groups of bacteria influence the flow of carbon and energy within and across ecosystems? And so we can think back to this sort of single cell and what it's doing and what its requirements are for growth and for survival. We can think about this at the metabolic level. We can also think about the interactions among organisms and how they're competing for resources and how their sort of environmental context all influences this. At a sort of central level, we can think about microorganisms as this input of resources that they use to fuel their cellular growth or their production. But to do this, they actually have to balance both the production of biomass and the respiration, often leading to the release of CO2. Most of my research focuses on heterotrophic organisms, so I'm sort of in this CO2 respiration category. And this might bring us back to these scary figures that you may recall from an intro bio class and you may try to actively forget about. Here on the left, we're showing the simple steps of glycolysis, and on the right, the simple steps of the Krebs cycle or the TCA cycle. And one important thing to realize in these is that we can calculate from this the theoretical energy yield. For example, the theoretical ATP yield from aerobic respiration. And you may remember from some table that you highlight every once in a while from Brock that from one glucose molecule, we expect about 38 ATP. But this is actually only a theoretical maximum if everything goes right. And also if we're only creating energy. But remember earlier I mentioned that cells need both energy and biomass. They're constantly balancing this production of CO2 via respiration, this energy creating mechanism, but also the production of biomass. And to build that biomass, they actually need to steal the intermediates from all of these earlier cycles, all the stuff that you actually didn't learn in your intro biology or perhaps your intro microbiology course. What cells do is they take intermediates, which I've highlighted here in red, as precursors to synthesize things like new amino acids. So to make any amino acid, we actually have to deplete the potential ATP yield for biosynthesis. So there's this constant balance between respiration, anabolism, and production, catabolism. And I often refer to this balance as growth efficiency. This is sort of how efficiently cells can grow, how efficiently they take those resources and they are able to create new biomass, but at the same time, generate the energy they need to survive and grow. And we can use sort of some simple calculations to calculate growth efficiency. So growth efficiency is just the production of is the proportion of biomass production related to the total carbon usage, which includes both production and respiration. Growth efficiency or variation in growth efficiency can influence the physiology and the growth dynamics of individual groups of microorganisms. For example, here on the right, I'm showing two just sort of models, bacterial groups. I labeled C1 and C2. And in group C1, they're producing proportionally less biomass than CO2, meaning they're growing pretty inefficiently, and they're serving as a source of CO2 to the atmosphere. And in population 2, labeled C2 here, they're growing more efficiently, so they're actually serving as a sink. They're actually contributing more to the burial of carbon in their ecosystem. So when these microbes are inefficient, they can release the majority of their carbon as CO2, and they can do things like promote an ecosystem that is a net source of carbon in the atmosphere. But when they become more efficient, or when we promote communities that are more efficient, those communities can turn into net sinks of CO2. And you might recall from some, I think you're in an aquatic focus group here, there's a lot of work across the US, Canada, and Sweden, people thinking of lakes and their heterotrophic bacteria contributing to lakes being a sink or source of CO2. So this is directly the cellular mechanism by which that happens. But we can step back and think, what are the physiological constraints that determine if an organism is growing efficiently or not? What does it mean or what constrains an organism to be in this source or sink category? And is there any taxon level variation constraining growth efficiency? And last, what is the role of the environmental context in which the organism finds itself in? So today I'm going to sort of outline this with 3 objectives. First, I'm going to use a trait-based framework to understand taxon level variation in bacterial growth efficiency. Then I'm going to compare growth efficiency across entire communities that range ecosystems in Quebec. And last, I'm going to develop a theoretical model to explain our observed variation. So the first bit of work, this traits-based work, comes from my PhD work, which was done under the supervision of Jay Lennon at Michigan State University and then Indiana University. And a lot of this incorporated the research of an undergrad researcher, Shumeng Howie, here in the upper right. You could find all this work in this environmental microbiology paper, which you can get to via this QR code at the bottom right. So for this trait-based approach, we used a collection of lake bacteria that I cultured from lakes in the Upper Peninsula of Michigan. You probably have seen Michigan, or people refer to Michigan as this like glove thing, but then there's this whole other region up here by Lake Superior. And these were all isolated from sort of these transitional lakes, these north temperate lakes in the Upper Peninsula, pretty close to Lake Superior. And from these lakes over a number of years, I isolated organisms that spanned a number of groups of bacteria, including alpha, beta, and gamma proteobacteria, which are the ones I used for this study. Using this culture collection, we measured bacterial growth efficiency when each isolate was grown using one of 3 different carbon substrates as their sole carbon source. So they were grown either using glucose, succinate, or complex carbon protocatechate as the only carbon they had for biomass synthesis and energy production. In addition to these growth efficiency experiments, we measured the maximum growth rate that each organism could grow in a complex media, and also its niche breadth by categorizing the number of carbon molecules it can use using the biolog ecoplates. For BGE, for growth efficiency, we measured this using real-time measures of oxygen consumption using the PreSense system, and continuous instantaneous measures of protein biosynthesis using a tritiated leucine assay. The niche breadth was calculated based on the number of total resources an isolate was able to use, standardized for the total it was queried against. So what we can first start doing is we can look at the variation in growth efficiency across our various isolates. Here I'm showing this, this tree in order to give us this trait-based approach, but also leave it in a phylogenetic context. And onto this tree, what we can do is we can map on our values for growth efficiency, which I'm showing here as a heat map type plot. From this, we can see that there's clear taxonomic patterns. For example, alpha proteobacteria are just not as efficient as gamma proteobacteria. But also the growth efficiency depends a lot on the resource that the particular organism is consuming. And we can see some really nice trends where for some organisms, they have the highest growth efficiency using things like glucose and the lowest growth efficiency using those complex carbon substrates. The taxonomic order throughout this analysis explained about 20% of the variation in bacterial growth efficiency across all resources. But across resources, species identity explained 58% of the variation in growth efficiency. So there's this really interesting dynamic where it matters on both who you are as far as the microorganism, but also what resource you have available to you. Then we can take that next step and ask if growth rate and niche breadth influence the efficiency. I'm going to wait to the end to answer the questions, by the way. To ask if growth rate and niche breadth influence efficiency. Here I'm showing a simple plot where I'm plotting growth efficiency along the y-axis and first growth rate along the x, and then Levin's index that measure niche breadth along the next plot. And what we can see is that for maximum growth rate, there ended up being two groups of organisms in our analysis that they grouped based on efficiency in this high growth efficiency group and a low growth efficiency group, which is a little bit more complicated, I can talk about later. But at least for the high efficiency bacterial growth efficiency group, there is a negative relationship between their maximum growth rate and their growth efficiency, meaning organisms that grew faster grew less efficiently. As far as the niche breadth or the number of resources they could use, we found no significant trend between the number of individual carbon substrates from the ecolog plates an organism can use and its growth efficiency. What we did find, and sort of the takeaway here, is that there's a strong negative relationship between growth efficiency and maximum growth rate. Isolates with a higher maximum growth rate have a lower growth efficiency. And our results from this simple regression predict that there's about a 2.6% decrease in efficiency for each minute increase in growth rate, at least for this high BG group. And I realize that this isn't a purely linear trend, that has to be nonlinear towards the extremes, but at least in this range in which we measured growth efficiency, we do find this sort of linear trend with this decrease in efficiency. So in this trait-based framework, we really showed that taxonomic identity matters for determining growth efficiency, but also resource identity matters. And a little bit more interesting is that faster organisms are growing less efficiently. Next, I'm going to take you to some work done during my second postdoc, where I was at the University of Quebec at Montreal, working under the supervision of Paul Del Giorgio and his Carbis group. And here the objective was really to compare growth efficiency across entire communities, ranging the sort of whole boreal ecosystem in Quebec. So this long-term project, Carbis, is the carbon biogeochemistry and boreal aquatic systems. And what has happened over a number of years is there's been measures of bacterial production and respiration in more than 350 lakes, rivers, wetlands, and beaver ponds across Quebec. The data set I'll talk to you about today ranges from 2010 to 2013. And using this data set, we asked how ecosystem type and environmental conditions drive bacterial growth efficiency. So the first thing I want to think about today is, what is the role of ecosystem type, or does ecosystem type really matter? So along the y-axis here, I'm going to plot measures of bacterial production, and then along the x-axis, I'm going to group them into ecosystem type, lake, river, wetland, and beaver pond. And from our data, what we observe is that ecosystem type has a strong impact on production rates, with lakes having, on average, a lower production rate than all of our other ecosystems, rivers and wetlands having a bit higher, and then beaver ponds actually having the highest rates of bacterial production. So how does this variation in production influence bacterial growth efficiency? Well, this is where things actually get a bit more interesting. It turns out that across all of our ecosystems, we observed higher growth efficiency in samples that had higher production rates. So ecosystems with the highest production rates contained microbial communities that are just more efficient. They're using their resources, their carbon resources, more efficiently here. And as an overall trend, we're seeing a positive but saturating relationship between production and efficiency. So what is driving this? We used a machine learning approach to to look at all of our metadata that was available to us and try to pick out drivers of this relationship. And it turns out that total nitrogen, which I'm showing here on the left, as well as things like phosphorus concentrations, have a strong positive influence on production rates. And interestingly enough, these relationships were stronger than the bulk organic carbon concentration or any composition of the organic carbon as determined by a pair-FAC analysis. So we tried to break down the composition of the organic matter available to the organisms, and that actually turned out not to be significant. So just to summarize that really quickly, we can get back to this in a second. Different ecosystem types have measurable differences in bacterial productivity, and that there was a strong positive relationship between production and growth efficiency. And this seems to be driven by ecosystem type, but also by nutrient availability, which seems to be a major player in these trends. But I just want to pause here and recognize that what I've shown you in these first two objectives are actually opposite patterns. In the first objective, we showed that when growth rate, another measure of productivity, increases, efficiency decreases. And in the second objective, we showed that when production, or essentially growth rate, increases, efficiency also increased. So to sort of get at this, I'm going to use a sort of theoretical model to try to explain this observed variation. So first, I'm going to develop a model based on the known cell volume scaling relationships. There's a simple relationship between cell volume and cellular mass. And you can get numbers for these values off of the online database BioNumbers. And using the cell volume and cell mass values, we modeled the sort of a simple volume to mass ratio. The empirical data, which is actually pretty sparse, is shown in the blue dots on the figure to the left. And we use this data to simulate a combination of new variables shown in red using this linear model. So we generated or simulated new values that have the same volume to mass relationship in order to have a little bit more data or parameter space in our models. Next, we used an energetic scaling relationship to determine how cell division time constrains growth efficiency. So the energetic scaling relationship model we're using comes from Lynch and Marinoff 2015, and it's really looking at the bioenergetic costs of a gene. And from this scaling relationship, we generated a model that demonstrates that there is a monotonic decrease in growth efficiency as cell division time increases. Essentially, and what is sort of the foundation for this, is that as cells take longer to divide, they actually need to use more energy to maintain their cells. So using this model, we can then model or determine how metabolism, specifically biomass production, constrains efficiency. So we have this same plot here where we have production along the x-axis and growth efficiency along the y. And what we're going to do is we'll use our model to generate some predicted values for growth efficiency based on various production rates using those cell volume, cell mass values we generated earlier. The red lines here represent individual models, and the blue is just the average among all of our models. And as a result, what we found is an increasing but saturating relationship between production and efficiency. Essentially, what's going on here, and sort of break down the math and make it more understandable, is that cells must use energy to meet their maintenance demands. It's sort of the fundamental part of cellular physiology, is that we need to maintain a viable cell. And therefore, growth efficiency represents the balance between that catabolic, that respiration, and that anabolic production. However, cells must meet that maintenance energy requirement, regardless of what they're doing or how fast they're growing. And therefore, efficiency will necessarily be low when production is low, because the maintenance part of the equation dominates. So when cells are growing really slowly, they're sort of approaching dormancy, they're still needing to use all that energy required for maintaining their cells, even though they're not producing any biomass. And this will therefore drive efficiency down. So to put our model in context, cells must meet their maintenance energy requirements, and therefore efficiency will always be low when production is low. As a result, they'll primarily respire organic matter to meet their maintenance energy requirements. And to put this in the context of our ecosystems, our sort of Quebec samples, in any energy limited system, our model and our empirical data demonstrate that increasing productivity, for example, adding nutrients to the system, will also enhance efficiency. And this is exactly what's happening with things like beaver ponds. But this was actually not the case in our lab experiments. Thinking about this sort of long and hard and trying to understand what's going on, we've sort of determined that our predictions, at least from our model, work best at the lower end of the growth rate spectrum. And what we think is happening is that other phenomenon are sort of are causing different situation at the other end of the production spectrum as it approaches its maximum. For example, in those growth rate experiments we did in objective 1, all those organisms were measured at their maximum growth rate in a complex rich media. And so we think that's happening is that things, when there's high growth rates, we think things like overflow respiration may be decreasing efficiency. Essentially, from a cellular mechanism, organisms can start producing, they can start fueling the TCA cycle so rapidly and sending essentially electrons through the electron transport chain that they're pumping protons across the membrane to create that proton motive force faster than ATP synthase can bring them back into the cell, so that we create a positive proton gradient outside the cell faster than the mechanics of the synthase gene can allow them to come back in. And this is called overflow respiration, and it leads to things like a decrease in the pH of the environment, but also a decrease in the efficiency of the organisms. All right, so now to sort of bring you to next steps going on currently in my lab. We're sort of moving these natural systems into the lab using some experimental chemostat experiments. So a chemostat is just a continuous culture vessel where you have a constant media input and you use a waste output to maintain the volume here at about 50 mils. And all this is done in a controlled setting where you can change the resource concentration of the media incoming, but you can also change the environmental context, like things like temperature. And we're using these to estimate the maintenance energy requirements of isolates, both from lake systems from Michigan, but also novel isolates here in Alaska, which are under a little bit more resource and energetic constraints. And we're going to use these chemostat experiments to test some of our model predictions, especially about maintenance energy dominating the equation. In addition to these physiological assays, we're going to start looking at gene expression profiles to understand what are the changes in gene expression as we change these resource concentrations and cause communities or sort of isolates to grow less efficiently. And we're also parameterizing cell death rate. You can look at our mechanism for parameterizing cell death rate in this PNAS paper that came out earlier this year. So to briefly put this all together, it seems like sort of using all this data, we're generating this bacterial growth efficiency framework where I'm simply showing this as a cartoon with production rate along the x-axis and efficiency along the y. And in a lot of energy limited systems like the lakes in Quebec, maintenance energy is high, and therefore as production rate increases, so will efficiency. But when we get into sort of some cultures which are growing in the lab that are growing closer to their theoretical maximum growth rate, we get some physical limitations and wasteful spending of energy that's limited by the sort of cellular biophysics of the cell. To put some conclusions for you, heterotrophic bacterial groups have distinct growth efficiencies that seem to be constrained by their physiological abilities. The resources available for growth and other ecosystem functions seem to regulate efficiency across both population and community scale. And then when systems are limited by resources, productivity is limited, and then maintenance energy requirements are going to dominate metabolism, driving efficiency down. And then as systems become more productive, efficiency increases, but there's probably a switching point at which systems become inefficient. To sort of bring this back to sort of my lab goals, by determining these physiological constraints, we're trying to better understand the role of bacteria in ecosystems and how communities and landscape changes influence ecosystem function. And thank you for your time, and I will happily answer any of your questions.